like to move to our last but not least speaker in this section. Let me give you Breno Fragomeni. Breno is a PhD in animal and dairy science. He's professor at Connecticut University, and he's participating in a project for studying genome by environment interactions and how they impact breeding values. That sounds difficult, but I believe Breno will decompose it for us in more simple terms. Today, he will shed light on this matter for us. Breno, are you with me? Can you hear me?
Yes, I can. Can you hear me?
Yes, we can hear you. Please let me check a bit with our translation director if your audio tracks are correctly set. Okay, good. Thank you. I see that you run the presentation and you can start when you are ready.
Okay, so, oh, I gotta make it full screen. Is it good now?
Yes, it's good now. We can see it in full screen.
So, first of all, thank you very much for inviting me for this talk. Genotype by environmental interaction. Why is it important to include local environmental factors in genomic evaluations? That's the title of my talk that was suggested. And it's a very good topic for me, because that is one of my main research interests here in Connecticut. So I'm going to talk a little bit about genomic selection and genetic gain, and then I will put the genotype by environmental interaction in context, trying to correlate how those interactions can impact genetic gain, especially when we ignore G by E. And I'll finish up with some recommendations for producers. So animals are different from each other. We look at two individuals, we collect phenotypes, there is going to be a variation. The reason for that variation can be on their genomics, on their genetics, and can be because of the environment too. However, the interaction between the two is also a factor that may make animals different. What we do, we try to use statistical tools, genetic and genomic tools, several linear models to try to identify out of the phenotype of an individual, what is because of their genetics and what is because of the environment. And then we select the animals based on their genetics portion. And that's what leads us to make genetic gain. How do we do that? Well, I'm going to give here examples in dairy. We have to estimate breeding values. They have to calculate breeding values. Initially, when the animal is born and we do not have genomic information, the only thing we know about the animal is the average of their parents. But we know that not all the animals from the same parents are going to be exactly the same, even though that will be our best guess. So that second term here is called Mendelian sampling. And that's what makes the difference between full sibs to not to be clones. How do we learn that a given animal is not just the average of their parents, that the breeding value of a given animal is not the average of the breeding value of their parents? Well, for a dairy bull, for example, let's call this animal here horns. We need to wait for this animal to grow so that we can collect semen, inseminate a cow. At year 3, the first daughter will be born. This daughter will be inseminated when this animal is 4 to 5 years old. 9 months for pregnancy, 305 days for the first lactation. Then we'll have an idea if our bull that's now 6 years old is a good bull or not. It takes a long time, but it's very accurate. That's what we call progeny testing, and that allows us to learn a lot about the Mendelian sampling, which will give us an estimated breeding value or a PTA with a high accuracy. The main problem here is that it takes a long time. And I'm going to explain in a second why this process being very slow is a big problem for us. Well, we can actually get into the same sort of model that will include the phenotypes, the pedigree, and our statistical model, and we can include genomic information. We can include DNA markers. Most of the times you're going to work with 50,000 SNPs. Sometimes we use a little bit higher density. There are actually examples that we use a much higher density, but 50,000 SNPs will be the most common type of chip that we use. And that will yield a higher accuracy, especially for the younger animals. And in a second, I'm going to show in an equation how important is that decreases generation interval. So we don't have to wait for an animal to be 6 years old to include that animal in our breeding program. It also allows us to correct pedigree errors. So if for whatever reason there is missing pedigree information or a wrong pedigree information, genomic information can help us to identify where the error is and sometimes even fix it correctly. It also allows us to understand more about the genetic architecture of the trait, which for now, it is mostly important for the research standpoint, but hopefully soon we'll be able to include that information into actual genetic evaluations and genomic evaluations. So I like formulas, and that is the form that I call the breeder's equation. So the breeder's equation measure this delta G here, which is the genetic gain per year that we observe. It is a consequence of selection intensity. So selection intensity is how intense our selection is. So the fewer animals we select, the higher the intensity will be. We cannot actually change it that much with genomic tools. We usually, with biotechnology for reproduction, mostly artificial insemination, that's the best way to increase selection intensity. However, accuracy of selection, the higher the accuracy is, the higher is the genetic gain. And that is something that we can gain a lot with genomic prediction. The genetic standard deviation is a characteristic of a population. We cannot do much, but we know that it also influences genetic gain. And most important for us is the generation interval. So of course, if our animal is very old, we are using old genetics, and that genetics being diluted through the years. With genomic selection, because we allow inclusion of younger animals, we are increasing genetic gain per year because we're being able to include younger animals with decreased generation interval with higher accuracy. So those are the 2 points that we're going to work with. So what about genotype by environmental interaction? Well, sometimes we have a good evaluation, but the results are not so good. Our prediction models basically fail. We select the best bull. We see in the field that the results are not what we expect. One reason for that could be genotype by environmental interaction. So different animals may respond differently in multiple environments. The best animal for a given place, country, region, it's not necessarily going to be the best animal for another region. Some animals are going to be very good in a very specific scenario and not so good in other specific scenarios. That can be a season. It can be animals that do better in the summer, animals that do better in the winter. They're not necessarily the same. It can be that animals are better. For example, in my research here, we are evaluating if the best animals for the region of New England, the Northeast USA, are also the best animals for California, because California has many more animals. Therefore, when we get a genetic evaluation, we get much more information from California than from here. So we are basically using animals that are better from other regions And we are not really sure if those animals are also going to be the best animals here. Diet, if we feed animals different diets, they may perform differently. The main thing with GBI is that it's not that animals are going to be affected by the environment, but when the rank of the animals change with the environment, that's when we get mostly concerned about GBI. So when animal for a given environment is not that is the best one in the first circumstance, is not the best one anymore, that's when we start getting concerned about GBI. How do we calculate that? We use the genetic correlation. In the previous presentation, we saw that we can use different populations that are as different traits, and that's exactly what we do when we study GBI in one of the approaches. So what do? We'll consider data collected in animals from a given environment. So in my case, one state to the other state, as there are two different traits. So milk production in New England is trait 1, milk production in California is trait 2. We see that a lot with Interbull, that they consider each country as a different trait. And there are many, many other examples. And then we calculate the genetic correlation between those two traits. It's going to measure how much of the genetics of one trait is explaining the genetics of the other trait. And it's going to vary between -1 and 1. If the correlation is negative and strong, so closer to -1, it means that as one trait increases, the other trait decreases. If the correlation gets close to 0, it means that the traits are not correlated. Therefore, we cannot learn much from one trait to the other. And when the correlation is positive, it means that increasing one trait will also increase the other trait, but not in a perfect scenario. If the correlation, and that's the rule of thumb, is less than 0.8, it may suggest that we need a two-trait analysis. We need to separate those two environments in two traits and have a specific evaluation for each specific environment. What are the problems of ignoring that genetic correlation? So there is this classic study in 1995 that they calculated genetic correlations between the US, Canada, and Denmark. The correlation between US and Canada was 0.95, which is very high. And the correlation between USA and Denmark was 0.84, which is still high. And what they did here, they compared the amount of animals selected in common on the top 100 sires, and they check what happened. How many of the top 100 animals were the same in each one of the countries when they considered GBI and when they ignored GBI. And what happened was that when they ignored GBI, the number of animals, especially between US and Denmark, decreased. So maybe we lost more than 40 animals in the top 100 that were not going to be ranked, which means that we may be including animals that are not the best and also ignoring animals that are the best because we're ignoring this very important value. That means that the best bull for Denmark for stature is not necessarily the best bull for the US for stature. And that is possible to correct if we have data for it. The other problem of GBI is that when it exists and we put the evaluations together, countries or regions with more data will overwhelm those with fewer data. And what will happen is that we may be selecting for the environment that contains more data. So splitting that into several populations that are genetically correlated will fix that. If we ignore that, we're going to select either the best animal for one specific region or the animal that is well-rounded and that will do okay in all the regions, but not necessarily the best among them all. In this table here, there was a simulation that we did to simulate a multiple across country evaluation, a MACE evaluation using genomic information. And we simulated the 4 countries with correlation with equals 0.8. And we use different methods to try to predict. And we see that the country with the higher number of genotypes, country 1, is not really affected by the method. However, the country with less genotyped animals is the one that has the most potential to learn from the other countries, but is also the country that has that will be really impacted if we ignore those informations. Over here, we have a table of several genetic correlations measured across traits in several different breeds, mostly milk yield, but you're going to see that as with departure from milk yield, so when you start talking about health, longevity, efficiency, fertility, the correlation decreases. So if we're thinking maybe just about milk yield, G by E is not a big concern, but when you start talking about health, reproduction, and efficiency, that's when we are going to start seeing those impacts. So this table here was made by my grad student that is actually pursuing the research comparing Connecticut and New England. What if G by E exists? We can rewrite that same formula as before, but now instead of using just the accuracy and so basically we transform that formula to include the genetic correlation. So how much gain is expected for a given trait if we select on the other trait? The two main things here are the genetic correlation and the accuracy. If the accuracy is very high and genetic correlation is high, genetic gain is going to be consistent across environments. But if genetic correlation decreases, then it doesn't matter if the accuracy is high, we're going to start losing information. So what are the numbers? Well, usually we talk about genetic correlation above 0.8. We not necessarily need to split the evaluation into two, but it depends. It needs to be evaluated. If one region has a much higher accuracy, it may be beneficial to include that region into the evaluation. However, in most cases, using data from different countries and environments will harm our decision-making, especially when genetic correlation is low, which is expected for fertility, health, longevity, efficiency, and so forth. So to conclude, management, diet, climate, and genetic base may cause re-rank of sires, and we call that G by E when that re-rank happens because of different environments. The best animal for a given environment is not necessarily the best for everywhere else. And as the genetic correlation for those traits decreases, the more different the best animals are going to be, which is going to decrease the rate of genetic gain. Using local evaluations is very, very often the best thing to do. And while the ideal scenario would be using a MACE evaluation, when it's not possible, local evaluations are often going to be the best approach. With that, I conclude my talk, and I'm happy to take any questions.
Thank you, Breno. That was really useful and interesting. Do you know maybe how many countries there are in MACE now that exchange their genotypes and evaluations?
I should know that number exactly, but right, I would say it's 18 countries, but I may be wrong.
Mm-hmm.
I should know that number. I'm sorry.
Oh, that's okay.
But yeah, I worked with MACE last time was like 3-4 years ago, and I actually look at it. So they have this huge genetic correlation table with all the countries. But it's around 20, I would say, if I'm not wrong. I want to say 18, but I may be wrong.
Okay, thank you. Around 20 is good enough for me. Thank you. Let me check in chats. Is there any other questions? Well, I believe no. No, not yet. Dear colleagues, if you have any questions, please ask them in the chats, and will ask Breno to answer them maybe later in the email. I think that we need to digest this information, because your speech was had a lot of bioinformatician terms. Thank you, Breno.
Thank you.
That was really great. Have a good day. I believe you have a morning there. Bye-bye.